the secret of Eve's apple is lifted. A team of international scientists decoded the genome of the apple, the largest plant genome uncovered to date. Coordinated by the Istituto Agrario San Michele all'Adige in Trento, Italy, the findings are now being published in the latest Nature Genetics issue. For 3 years, the scientists studied all 17 chromosomes of the Golden Delicious, the world's second most widespread domesticated apple. Dr. Silvio Salvi was one of the specialists in the international team. It is interesting to note that the apple has doubled its genome. This duplication happened 50 million years ago. This duplication of the genome of the apple complicated the sequencing process. But with the help of state-of-the-art technologies, the team decoded all 57,000 genes. Now this knowledge helps to improve the molecular-assisted breeding strategies. First, the parents of the cross are chosen on the basis of the genes which may contribute to a given characteristic, like color or crispness. Then the scientists study the genes of the seedlings and identify those with favorable gene combinations. The selected apple seedlings are then planted on the testing fields of the Institute. From the thousands of different varieties, the scientists choose the best for further examination. Dr. Ricardo Velasco was the main author of the Apple Genome Project. Clearly, wild plants growing in those areas where pathogens have developed contemporaneously develop some resistance that domesticated plants obviously do not have, because they are protected by humans. These wild plants containing resistant genes can be used as parents in the gene crossing programs. The origin of all apples lies in Kazakhstan, where they're still growing. For the cultivated apples, the most favorable genes lie in their ancestors' genome. In the wild apple varieties, the scientists found 992 genes responsible for disease resistance. This resistance to fungal or bacterial pathogens can now be used to breed new apple varieties. The Istituto Agrario San Michele has a long tradition in combining traditional breeding techniques with modern genetics. We've helped breeders who try to identify new varieties to create new plants of the future, starting with very precise objectives. They now know, for example, the genetic base for resistance to pathogens, different quality features, crispness of the apple, and the conservation characteristics. All these are features which finally lead to the product the consumer is asking for. The apple is the main fruit crop of the world's temperate regions, and so far 10 apple varieties dominate the market. In the future, we may buy apples which need less chemicals and are healthier and tastier, thanks to the finding of the apple's genome.